In the previous talk, we talk about how the plant pathogens, despite tiny and fragile, are capable of causing disease in the plants which are much bigger and stronger. Uh, Today, we are going to talk about how the plants defend against the pathogens despite having certain disadvantages. The first disadvantage of the plants against the pathogen is the number. The number of the pathogens which are capable of causing disease in the plant is in thousands. So plants must have the strategies to counter these thousands of pathogens to remain viable and live in the field. The second disadvantage associated with the plants against the pathogens is that pathogens not belong to a single species. Rather, they are a diversified group of individual. For example, the fungi are eukaryotes, the bacteria are prokaryotes, nematodes belong to kingdom Animalia, while the viruses are the separate taxonomic entities along with the viroids, molecules, and fungal-like organisms. All these pathogens vary when it comes to their taxonomy, size, shape, mode of attack, reproduction, and distribution. So the plants must have strategy to counter each and every one of them in order to survive in the field, which is a huge task. The fourth disadvantage associated with plants against the pathogens that unlike the human and animals, plants are sessile. They can't run away from their enemies. And you are just seeing that the whole human race is paralyzed against the COVID-19. Despite so much advancement in the science for last few decades, along with that lockdown strategies, but we don't find any answer against this virus. And now look at the situation of the plants, which are at the mercy of the nature, how they can counter so many pathogens which are ready to attack on them. The last one is that in comparison to the other disciplines of the biology, such as microbiology, zoology, botany, plant pathology is relatively a young discipline, and it is just 150 years old. However, in last 150 years, so much has to be done to understand about the basic ideas or the basic facets about the plant pathogens and the plants and the environment, but still there are many things which are not completely understand that why the plant defend themselves and why the pathogen attack on them. So, do plants stop growing in the field despite so many disadvantages? The answer is no. The plants are happy and healthy, and we are seeing them all around us, and they are giving us food and shelter and medicines. So, it means that the plants has some kind of natural capabilities or the qualities which help them to remain protected against the pathogen. So, how plants defend against the pathogen? This is the topic we are going to discuss in the coming slides. Plant resistance works in layers, and the outermost layer is structural in nature. The nature provided the plants certain structures which not only help them to remain protected against the adverse environmental conditions but also against the pest attack. Why the pathogen attack the plants? Because the plants carry the nutrients, and these nutrients are present in the cytoplasm. So ultimately, every pathogen desperate to reach the cytoplasm. But in order to reach the cytoplasm, it has to come across the different kind of layers, which may be wax in nature, maybe cutin, cellulose, pectins, then plasma membrane, and finally the cytoplasm. And most of the pathogen don't have the ability to come across or pass these all these layers and finally to reach the cytoplasm. So the plants remain protected against most of the pathogens. Why? Because they have certain structures such as wax layer, cellulose, pectin, and hemicellulose, which the pathogen can't breach, and they remain protected against the pathogen attack. Sometimes the plant defend against the pathogen because of the presence of phytoantispins. Phytoantispins are the pre-existing antimicrobial compounds which help the plant to defend against the pathogen attack. These antimicrobial compounds are part and parcel of the plant physiology, and whether the plant is attacked by the pathogen or not, these chemicals are produced, and additionally they protect the plant against the pathogen attack because of their antimicrobial nature. You all know that the onion has two kind of varieties. One produce the white color bulb, while the other is going to produce the red color bulb. And it is noticed that those varieties which has the reddish color, they are resistant against the disease which is caused by the fungi, because these red varieties produce phytoantispin which is known as catechol. As the catechol is not produced by the white varieties, so they are always remain susceptible against the smudge disease. So here the presence of the antimicrobial compound help the plants to defend against the pathogen attack. Sometimes the plant remain resistant because they produce certain kind of antimicrobial compound in response to the pathogen attack. These chemicals are known as phytoalexins. Due to the production of the phytoalexin, the plant counter the pathogen. The phytoantispin and phytoalexins are same because both of them are antimicrobial in nature, but the only difference is that the phytoantispins are produced whether the plant is attacked by the pathogen or the not, while the phytoalexins are only produced in response to the pathogen attack. For example, the researcher observed that those pea varieties which are able to produce picetin, that is phytoalexin, remain resistant against the blight disease, and the varieties which are unable to produce the picetin are infected by the blight disease of pea. The plants defend themselves indirectly. Why? Because they don't produce the nutrients which are required by the pathogens. And pathogens are very selective when it comes to the nutrients. They can only attack those plants or those varieties of the plants which produce the certain nutrients. If those varieties are not producing that certain nutrients, the pathogens usually avoid them and not attack them. Here are the two examples which we come across in our daily life. First is the black scarf of the potato. This disease is caused by a fungi which is known as Rhizoctonia solani. And if remember, or even today, if you wash your potato tuber, you can find these outgrowth black color fruiting bodies of the fungi, which can easily be removable with the nail of your finger. So this actually a disease potato which is under attack of Rhizoctonia solani. But this Rhizoctonia solani fungi is very selective when it comes to the nutrients. It only attacks those potato varieties which provide the specific nutrient, and if that nutrient is not present, the pathogen will not attack on it. The other example which we come across is the apple scab. Here, some portion of the apple fruit become dead, which can easily removed when you cut it a little bit deep, and this disease is known as apple scab. Again, this fungi, which is known as Venturi inequalis, can only attack those apple varieties which produce certain kind of nutrients, and if these nutrients are not present, the pathogen will not attack. So nutrient is one of the factor which help the plants to defend against the pathogen. Kind of the resistance which is due to the presence of some structures or the presence of some antimicrobial chemicals like phytoantispins or the phytoalexins or the unavailability of certain nutrient is known as non-host resistance. Non-host resistance is defined as when an entire plant species is resistant against a specific parasite or a pathogen. And the non-host resistance is the most durable and the common form of the resistance. Why we use the word durable? We never heard that the cotton leaf gall virus, which is a pathogen, start attacking the nearby mango plants. So it means the cotton is the host while the mango is the non-host, and for thousands of years the mango is still the non-host of the cotton leaf gall virus. And same is the situation. The both brassica and wheat grow side by side, but it never happened that the wheat rust fungi start attacking the brassica for so many years, maybe for hundreds of years. It means why? Because the brassica is the non-host of the wheat rust. Why brassica is non-host? It may be due to some structure, maybe due to some production of phytoantispin or phytoalexin, or maybe brassica don't have the nutrients which is required by the wheat rust fungi. So non-host resistance is the first layer which help the plant to remain resistant against the pathogen attack. The second layer of the plant defense system is known as host resistance. Host resistance is defined as the resistance shown by the host species against the particular pathogen. For example, when a wheat variety such as zinc shows resistance against wheat rust, we say it is due to host resistance. How host resistant works, it is elaborated in the coming slides. If you look at the screens, you can find two bacteria at the top. These two bacteria are quite similar in size, in shape, and in every aspect. But one bacteria we call a virulent pathogen, and the second we call the virulent pathogen. At the bottom, you can find two host cells, which are again same in size, shape, in every aspect. But one we called susceptible and one we called resistant. Why it is so? We call the bacteria a virulent pathogen because it produces the protein which is known as elicitor. We call the bacteria a virulent pathogen which again produces the protein, but this time we called it effector. Why we called it the protein of a virulent elicitor? Because it elicit the resistance reaction when it interact with the resistant protein. In while the why we called it effector protein in case of virulent pathogen, because this protein can easily cause the disease both in the susceptible plant and in the resistant plant. How it happened, this comes in next slides. First, we discuss the case of a virulent pathogen. And if you recall, a virulent pathogen is a pathogen, and except that it produces a protein which is known as elicitor protein. What will happen when this a virulent pathogen attack a susceptible plant? The susceptible plant doesn't have any kind of resistant protein, and the end result is disease because there is no protein to detect the a virulent protein that is known as elicitor. But when this try to attack a resistant plant, this time this protein is detected by the resistant plant and a resistance is occur, and this pathogen become avirulent. So the virulence of a pathogen is dependent upon the host reaction. If host reacted, it make it avirulent. If host don't detect it, it remain virulent. Now, what will happen when a virulent pathogen tries to attack a susceptible host? The virulent protein goes right inside the host cell where it causes the disease because the host cell doesn't have any kind of resistant protein to detect it. So the end result is disease. What will happen when a virulent pathogen tries to attack a resistant plant? In the previous slide, we saw that the resistant protein efficiently detected the avirulent protein or the elicitor protein and make the plant resistant. But this virulent or the effector protein dodges the resistant protein and enters inside the host cell and cause the disease. At this point, I would request you to go through 3 or 4 times these slides so that you have a better idea about virulence, susceptibility, and resistance, and about their interaction. Here is a self-explanatory checkerboard which is showing the interaction between the pathogen and the plant. At the top, you are seeing the plant with R or the resistant gene that and the plant with small r without the resistant gene. Then on the other side, you are seeing a capital A, which means the pathogen is avirulent, and at the bottom, the small a, which is showing the pathogen is virulent. What would happen when these plants and the pathogen interact with each other? Suppose a virulent pathogen tries to attack a resistant plant. So the resistant plant has the R gene, and that R gene is able to detect the elicitor protein, and the end result is resistance, which is on the checkerboard shown by minus sign. Now, the avirulent pathogen tries to attack a plant without the R gene. It is a pathogen, remember. So, when the plant unable to detect the pathogen, it means that there is disease, and that is shown by the plus sign. The other case is that the virulent pathogen is trying to attack two type of plant. One is resistant and the other is susceptible. When it is attacking the plant which has the R gene, but the virulent protein or the effector protein has the ability to dodge the R protein, the end result is disease, which is shown by the plus sign. And now this virulent pathogen, which has the effector protein, tries to attack a susceptible plant which doesn't has any kind of R protein. So obviously there is disease, and it is again shown by the plus sign. Always remember that whenever the resistant cell able to detect pathogen inside it, it opt for the suicide. Not only to kill itself, but also the pathogen present in it as a result killed. This reaction or response is known as hypersensitive response, which is only shown in the host resistance. During the beginning of this course, we came across work of Alvin Stackman, who in 1915, while working on cereal rust, first time used term hypersensitive response or HR. He defined it hypersensitivity of plant towards rust fungi. He noted that the area showing HR became dead. He also found a correlation exists between HR and resistance, and those varieties which showed HR are resistant one, while those which showed no HR were susceptible one. HR is quite regularly used in the plant pathology laboratories all over the world, either to assess the resistance of the plant variety or to assess the pathogenicity of a microbe. Here you are seeing a plant pathology student injecting a unknown bacterium inside the tobacco plant to see whether it produced the HR or not. And on the right side, you categorically can see a dead necrotic brown colored spot appear where the bacteria is injected, while the portion of the plant which is injected with the sterilized water remain green. So it means that the bacterium injected here are plant pathogenic, and secondly, the tobacco plant is either resistant or non-host to it. But the important thing is also here to note that the HR is confined to a certain area. That is why HR is also known as localized acquired resistant. Why we use the acquired? Because this resistance is due to the injection of the microbe. If the microbe is not injected, then the plant would not exhibit any kind of resistance. Frank Ross was a teacher and a researcher in the Cornell University, and after the 36 years of Stackman discovery of HR, he performed an experiment on the tobacco mosaic virus in order to see the impact of HR on the overall resistance of the plant. So he used another term which is known as systemic acquired resistance, which we are going to see how it works. I try to elaborate the work of Frank Ross by this simple picture. On the left side, you are seeing a tobacco plant, which has many leaves, one, two, three, four, five. And when this tobacco plant was inoculated with a tobacco mosaic virus, and if you look at that, I gave it a black color. So what happened that this tobacco plant showed the disease symptoms, which are the appearance of mosaic area. It means that the strain which is used in the inoculation was the virulent one, and the tobacco plant was susceptible against it. But in another plant, the Frank Ross changed his strategy. This time he inoculated the lower leaf of the tobacco plant with another viral tobacco mosaic virus strain, and I gave it a green color. So it is again tobacco mosaic virus, but it is different from the first one. What happened next was the production of HR. And from the Stackman work, we know that only those varieties are resistant one which gave HR. So it also means that this strain of the tobacco plant, which is shown written in the green letter, is the avirulent one, and the tobacco plant against it is the resistant one. That is why it is producing HR. But interestingly, what happened next? That on the same plant, he inoculated the strain which is the virulent one and against which this tobacco plant is susceptible one. But this time, the disease is not occurred. It means that now this plant is not only resistant against the avirulent strain, but the virulent strain of the same virus. And he repeated this experiment by first inoculating the avirulent strain to the upper leaves and inoculating the lower leaves with the virulent strain, but he got the same result that every time the tobacco plant not show resistance against the avirulent strain but also the virulent one. So from that he inferred that this resistance which is activated in this tobacco plant is actually somehow related with the localized acquired resistance or HR, which not only confine the pathogen but also make this plant systemically resistant against the virulent pathogen. And he used the term systemic acquired resistance. And interestingly, he also noted that this resistance remain viable not for 1, 2, or 3 days, but up to 20 days. It means that if you initially inoculate the A virulent strain, the plant remain resistant against the virulent strain not for 1, 2 days, but for the 20 days. Another finding of the Frank Ross was that this resistance is not only effective against the same strain of the tobacco mosaic virus, but this plant is also become resistant to other strain of the viruses as well as against the pathogenic fungi. So the systemic acquired resistance is not only effective against the same strain, but it is also give a generalized kind of resistance, and plant once the systemic acquired resistance activated in it remain resistant against couple of pathogens. So because this resistance is not only confined to a specific area, like in case of HR, they use the or he used the term systemic acquired resistance. So from that one, I think every one of you now understand what is the systemic acquired resistance. Secondly, if you little bit compare it with the vaccination which is going on in the animals and the humans, in case of vaccination, we were given some kind of polio vaccine in our childhood, or the vaccines of the measles or the TB, and our body remained resistant for the rest of our life. So here the concept is almost same, but we not use the word vaccination. Why? Because the plants don't have antigen antibody reaction, and secondly the plant defense system is not circulative. It don't has any kind of T cells, white blood cells, or the memory cells. So in plants we use the word systemic acquired resistance. But if the similar kind of phenomena, if it occurs in the animals and the human, then the word vaccination is used. So if we want to define what is the systemic acquired resistance, it is the systematically activated resistance after primary infection with the necrotizing pathogen. This resistance is different from the non-host resistance as it is activated by some avirulent pathogen. It produces the symptom, but most of the time it is not observable in the field. Its symptom in the form of HR, but it is so minute and tiny, but we can hardly see it in the field, and that is why we don't realize that the plant are exhibiting systemic acquired resistance while they are growing in the field. But every plant exhibits systemic acquired resistance. That is why it remain viable and live despite the presence of so many virulent pathogens in the field. Suppressive soils are those soils which have antagonistic microorganism or the biocontrol agents against the plant pathogens. So these are the soils which naturally have these microorganisms which not only killed other harmful microorganisms, but when they are present in the rhizosphere, they make the plant resistant against these harmful plant pathogens. These beneficial microorganisms are also known as plant growth promoting rhizobacteria or the fungi, and they are abbreviated as PGPR or PGPRF. The major genera belongs to PGPR include Pseudomonas, Bacillus, and Trichoderma. The above two are the bacteria, and the Trichoderma is the fungi. In the later part, the scientists applied these microorganisms into the rhizosphere, and when the plant's root interact with these microorganisms, the whole plant become resistant against the harmful pathogens. Such resistance is known as induced systemic resistance, or it is also known as ISR. ISR or the induced systemic resistance can be defined as a systemic resistance in the plant that is triggered by certain strains of non-pathogenic root colonizing bacteria. It is a symptomless, unlike the systemic acquired resistance where HR produce a necrotic area. Many of you ride bikes and have a better idea to protect yourself against any unfortunate incident. You wear the helmets. Similarly, in its life, the plant is attacked not only by one pathogen but thousands of pathogens which vary in their shape, size, mode of nutrition, mode of attack. So, according to that, it has to wear many helmets or many shields. So the outermost shield or the layer is known as non-host resistance, and most of the pathogens are filtered at this layer. And then there is a very specific another shield which is known as hypersensitive response, and that is a form of host resistance. That hypersensitive response not only confined to a certain area but it is extended throughout the plant. Then we call it systemic acquired resistance. And there are many beneficial microorganisms are present in the rhizosphere of the plants which not kill the harmful pathogens but also make the plant resistant against the different kind of harmful pathogens. So that resistance is known as induced systemic resistance. So actually there are four different kind of layers help the plant to resist against the thousands of the pathogen. And you always know that the plants are also attacked by the insect and the herbivores. So in order to protect against them, they have another shield or the helmet which is known as herbivore induced resistance. So all these different form of the resistance work independently and jointly, but their goal is to protect the plant against the harmful pest. So I hope that at this point everybody has a very brief and good idea how plant defend themselves against the plant pathogen. So I would like also to conclude my talk and have a nice day and goodbye.